Good morning and welcome to the third webinar in this series dedicated on COVID-19 response in low and middle income countries. A series that is co-convened by the African CDC and FHI 360. My name is Dr. Otto Chabikuli, Director of Global Health Population and Nutrition Business Unit at FHI 360. And today's panel will discuss all things COVID-19 vaccine research from a Global South perspective. You will notice that the chat function was disabled as precaution against possible Zoom bombing of this session. However, we welcome your questions via Twitter at the address shown on the screen, hashtag AskCovidExpert. I look forward to receiving your questions and I will submit to the panel at the end of the webinar. So let me just repeat again the hashtag for the Twitter account is hashtag AskCovidExpert, and we will be collecting the question as they come in. Before we proceed, I would like to read an excerpt from the public statement posted on June the 5th by Patrick Fine, the CEO of FHI 360. Can I have the public expert posted on? All right, while we're waiting for the slide to be put up, we can start the conversation. I'm joined today by a very expert panel, distinguished guests, Professor Quarraisha, Abdool, Karim, and Dr. Tim Mastro. Professor Quarraisha Abdool Karim is an infectious diseases epidemiologist and Associate Scientific Director of Quarraisha. She is also a professor in clinical epidemiology at Columbia University in New York and Pro-Vice Chancellor for African Health at the University of KwaZulu-Natal in South Africa. Over her long and illustrious career, she's received many awards, including the African Union Kwame Nkrumah Prize for Science and Technology, and more recently, the John Dirks Canada Gairdner Global Health Awards. Quarraisha, thanks for joining me today.
Thank you, Otto. It's a real pleasure to be here. And also, I want to thank FHI 360 and Africa CDC for their insight in convening this webinar and disseminating information that we so badly need to access as we learn more about COVID-19. So thank you.
And Dr. Tim Mastro, he's the Chief Science Officer at FHI 360 and also Adjunct Professor of Epidemiology at the Gillings School of Global Public Health, University of North Carolina at Chapel Hill. Prior to joining FHI 360, Tim served in various scientific leadership positions at the US CDC. He refers to it as a quick 20 years, it passed so fast. Tim is recipient of numerous awards at CDC and a prolific scientific author in peer-reviewed journals. Tim, thanks so much for joining us.
Thank you, Otto, and I'm delighted to join my friend and colleague Quarraisha on what should be an interesting webinar. Thank you.
Great. So Quarraisha, 8 months ago, many of us had a life. And that life had routines like, waking up in the morning, driving to the office, on the way you grab a cup of coffee, and on a weekend, shopping, having dinner with friends and family. That's just about 8 months ago. What we're living today has no resemblance to that. And for most of us, going back to that lifestyle, the only thing that is standing between us and that lifestyle is to have an effective vaccine. So the search for the vaccination is on, and the whole world is invested billions of dollars to get the vaccination. This pandemic started in the global north, Asia, North America, Europe, and so on. And Africa seems to have been spared at this point in time somehow. So we do have time. Now, the question is, while Africa has this luxury of time, what should be the role of Africa in the race to finding an effective vaccine? What is your opinion on that?
So Otto, that's an excellent question. And you know, while you say you were saying 8 months, I kind of think about it more like 6 months. 6 months ago, or even 5 months ago, we never knew our world was going to change like the way it has, and it has indeed changed. And sometimes when I think about it, I'm thinking, wow, you know, we're going to sit with our colleagues maybe in 4 or 5 years' time, hopefully. And then we're going to say, you know, in that time, in ancient, it almost will be a bygone era. But I'm an optimist, and I definitely think that that we, despite the devastation in terms of loss of lives, despite the disruptions that we've seen socially and economically, even as we grapple with the public health response, there are many, many positives in the COVID-19 epidemic. And I think one of them in dealing with the pandemic, and as we say, building the ship as we're still sailing, is that global solidarity comes through very, very strongly again. It is refreshing to see something where Africa is not number one. And I always like Africa number one when it comes to soccer or rugby or cricket. But as it comes to diseases, we know, you know, the standard thing with HIV is, well, 1 in 5 infections that are taking place is in young women, maybe even 1 in 4, and that 70% is in Africa. And for the first time, we're actually seeing that Africa has, I mean, there isn't a country on the African continent that doesn't have COVID-19. So we very much part of the pandemic. The question is, when will we reach the surge that we're seeing in some of the countries in the Northern Hemisphere? And hopefully, before we reach that point, we will have vaccines, or will be at a more advanced stage, so we don't see the mortality rates and the economic devastation that potentially could result from an uncheckered spread of COVID-19.
Thank you very much, Quarraisha. Tim, any opinion, any thoughts about that?
Well, I also tend to be an optimist, and so I cut my teeth as an early scientist investigator on the HIV/AIDS epidemic. And what encourages me about COVID-19 is the comparison of the speed of the science. For HIV, recall, it took us 2 years from the first cases of AIDS to identify the virus. With COVID, within a few weeks of the first cases of disease, we had a full-length sequence of the virus. Within another month, we had very good serologic assays, PCR assays. So that happened in the course of 2 months. For HIV, it took 4 years from the first cases to the first serologic assay. And then it took us another 15 years from the first cases to get effective therapy for HIV. And now we have a couple of drugs that are already showing some efficacy against COVID. So the pace of science is extraordinarily fast. The amount of energy that's being put into COVID vaccines is overwhelmingly impressive. For HIV, after 37 years since we found the virus, we still do not have a safe and effective vaccine. For COVID, I think we'll do far better. And today we can talk about the landscape, where the science is going. And I would just say, in those 40 years from the advent of HIV to the advent of COVID, the scientific field is light years ahead in terms of capacity on virology, immunology, and vaccine science.
Now, before we go even further in our discussion, I want to go back to the public statement that Patrick Fine has posted on June the 5th, talking about how our world has changed. There are other things that have happened in the past several months that remind us as human beings of the value of lives, Black lives in particular. And in that statement, what FHI is committing to is that we acknowledge that with programs in the United States and over 60 countries around the world, we pride ourselves on being part of the fabric of the communities where we live and work. In the United States, however, our Black and brown citizens experience relentless injustices, indignities, and frustrations that many others in America do not or have learned to ignore. As an organization accustomed to working in fragile states, this week we acknowledge that our headquarters is located in a fragile country. We stand in solidarity with all people who repudiate racism, misogyny, and all forms of discrimination. Signed by Patrick Fine, CEO of FHI 360. Thank you very much. So, Quarraisha, if you could tell us a little bit more about the epidemiology as seen from the Global South, particularly Africa, and the roles of partnerships that we can bring to bear.
Sure, and just to say that, just reflecting on Patrick's statement, I think that any form of discrimination weakens all of us, because we are one community and one human race. And as soon as we are one portion, whether it's by age or race or ethnicity differentiated and disadvantaged, all of us are shortchanged by it. And I think coming from South Africa, we know this only too well. And I think on the African continent, we've seen between colonialism and other forms of discrimination, how that can really undermine development and full contributions that people can make. So to look at the epidemic, and I think it's really important as we think about a vaccine, that we look at the magnitude of the problem, we understand characteristics of the epidemic, etc. And if we look in Africa, as of the 1st of July, we have seen just over 10 million cases globally and 500,000 deaths. In Africa, we have seen about 3% of the global burden of infection, just over 400,000 cases and about 10,000 deaths. And I think if we look in some of the states in the US, this is the numbers you will see in one or two states, and you can see from the size of the bubble here, what is important in terms of understanding the numbers is that there isn't a country not impacted and affected, and that any advances that we make in terms of stopping the spread of the disease or in terms of mitigating illness will benefit all of us. May I have the next slide, please? What we've seen is that the rate of transmission is very much dependent on how the virus is introduced, what are the bridging populations, and that establishes infection in the community. And I think that the goal in many African countries of flattening the curve has been really important and very early on in our response, by stopping public gatherings, by stopping international travel, and closing schools and introducing national lockdowns. We were able to contain the introduction of the virus in many of our countries, and thereby also reduce the number of individuals like healthcare workers and contacts being exposed to the virus and slowing the spread of the virus into our communities. But notwithstanding the fact of the success of national lockdowns, without natural immunity or a vaccine, almost everyone may be at risk of the coronavirus infection, and increasing numbers may need hospital care. So this is a really good public health goal. But we also know that Africa has large numbers of people who are unemployed and are socially and economically vulnerable for a whole range of reasons. So as we continue to respond to the epidemic and build on the success of the first 3 to 4 months of the virus on the continent, we have to start juggling and balancing how do we save lives with the public health responses, but at the same time, how do we ensure that we don't further exacerbate economic hardships and some of the structural challenges that face countries and economies where more than 50% of the population is dependent on day labor and on gray informal sector economies. So vaccines become really important in that context. May I have the next slide, please? So while we're waiting for a vaccine, and we're going to talk about and hear a lot more about vaccines, what we do have already available, I like to think about it as today's vaccines, and that these interventions that we have, things like social distancing, things like hand hygiene, cloth masks, symptom screening, appropriate use of personal protective equipment, testing, contact tracing, etc., while individually may have some benefits, in combination and where it's used in terms of large coverage rates, we can have much more impact using what we already have while we're waiting. May I have the next slide, please? Now, some of these behaviors to adopt are quite challenging. In fact, quite often we see in public health, we have the interventions, but we have low uptake of those interventions. And I think COVID-19 is again highlighting some of the issues on how do we translate the tools that we have in terms of adoption and adherence in vulnerable populations. But I think in terms of the SARS-CoV-2 epidemic, an additional issue, and in terms of control and containment, is understanding immunology and immune responses. And I think I want to highlight a few challenges that we are currently facing in terms of stopping the spread of the virus, and that will also impact and inform vaccine development. The first is that we have this pre-symptomatic phase where people continue to spread the virus without even knowing it and before they get ill. Then we have a whole other group, and this seems to be quite a large group of asymptomatic individuals who are spreading the virus without even knowing that they infected. And then we have those individuals who are able to what we call super spreaders. And I think the classic example is patient 31 from South Korea, who over a matter of days infected over 1,000 individuals. And then the other issue is that the virus spreads rapidly. And we know for every infected person, at least 2 others can get infected. And then the last point, which is we really don't know what the trajectory of this virus is going to be like, either globally or at a country level. So even at a country level where you can have initial control, you have this ever-present risk of new epidemics. And this can take many shapes and forms. With other respiratory illnesses, we've seen recurring small outbreaks. We've also seen, like with the Spanish flu, this monster wave, 50 million deaths, and then some slow waning of that. And then will we see what appears to be like, in the absence of a vaccine, a persistent crisis? We keep having infections taking place and the outbreaks at different places. May I have the next slide, please? So one of the things we think about when we think about vaccine development is the need for herd immunity. And in the COVID-19 context, it's a challenge to define what herd immunity actually is. And when we think about immunity, there's immunity that arises from natural infection, and then there's immunity that can be induced with the vaccination. And so when we think about vaccination and herd immunity, there's issues that we know from other respiratory illnesses. And I even am embarrassed to say that what is that threshold we need to be reaching? And for some things we can say 10%, and for others 90%. For COVID-19, it's a real black box. But what we do know is we need a vaccine, and Tim's going to talk a lot more about the vaccines that we have in development. But in finishing up this piece, I'd like to end with this last slide, which is saying unquestionably, Africa, as the rest of the world, needs a vaccine. And that while Africa has been a bit slow, may I have the next slide? in terms of developing vaccines, and there's a whole history there around infrastructure and investments and innovation needed, there are still other things that Africa can do to contribute to vaccine development. And in evaluating what of the 150 and growing numbers of potential vaccines there are, is how do we know which vaccine is appropriate for Africa? And so Africa can, and African countries can know this by understanding the characteristics of the evolving viruses locally and comparing that with what is happening globally. We also need to understand what happens once somebody gets infected, what is, in other words, etiology and pathogenesis, and how is the etiology and pathogenesis in Africa different from other places. In order to know who is infected, we need big and strong and reliable diagnostic testing capacity. We're already seeing in Africa how that is a challenge. We're having problems with securing reagents. We're joining the bottom of the queue in terms of diagnostic tests, and we want to make sure, one, we build the diagnostic testing capacity, but on the other hand, we don't want to be at the bottom of the queue when we have an effective vaccine. And these are our contributions. We have to understand our epidemics across the continent. What are the trends? As we noted, the introduction of the virus and the growth and magnitude of it has varied across the globe in countries, some countries being able to contain it, and others where it's just been like a rampant fire spreading very, very rapidly. So in the context of vaccine development, knowing your epidemic and knowing it at a country level within a country is really important in terms of the timing of trials and establishing those areas that are starting to see high incidence rates so that we get answers quickly with smaller numbers of individuals. We also have to have local data on circulating viruses and what the immune responses are and what they mean. Because right now, our understanding suggests that if you have severe disease, your immune response is much stronger than if you're asymptomatic. And with asymptomatic infection, we seem to have a waning immunity. So all of those are important. A big plus in Africa is the clinical trial capacity that has been built over the past 2 to 3 decades in response to epidemics like HIV, TB, Ebola, malaria, etc. But the regulatory capacity is uneven and patchy across Africa. It's important that we strengthen that. We also have a history with HIV, TB, and Ebola of engaging communities. And the more we engage communities, the better prepared they are for participation in trials, and the greater the acceptability are for interventions, because knowledge is an important prerequisite. And then the last point is that as Africa thinks about vaccine and vaccine development and their contributions and partnerships, we also need to start thinking about manufacturing capacity and post-trial access so that we don't end up with a safe and efficacious vaccine, but we actually don't, we again waiting in line at the bottom of the queue in terms of access to something that already works. So let me pause then. Otto, I'm happy to take any questions and discuss this further. Thank you.
Thank you, Quarraisha, for such a succinct description of the context and laying really the scene for us. I like, you know, there is having the scientific facts and knowing the science from an African perspective is one thing, but conveying it to the citizen in a language that they will understand so that you bring them on board, talking about the stakeholders and community mobilization and ability and readiness for them to accept the vaccine when it will be ready is another. I mean, just a few months ago, it just took a couple of French clinicians on a program, TV program, to almost light a fire by just some maybe poorly worded assessment of what should vaccine trial look like in Africa. What is your opinion about that particular element of communication and translating science as you know it into a language that will prepare the population to receive vaccines?
So I think that, you know, when you put Paul Fine's comment up, that excerpt, it reflects a sort of othering that goes on. If you don't look like me, you don't talk like me, it's almost dehumanization that happens. And I think in Africa, we have a long history of that, of exploitation, etc. In addition to the French comments, very recently, we've had a trial of BCG and its potential of preventing COVID-19 in healthcare workers stopped, just because people don't take the time to actually take the public with them and key decision makers. And it's like, you know, there are a few things. The one is communication is important and transparency. And I think in the conduct of HIV, TB research, and particularly with the Ebola outbreaks, it's amazing how it took many epidemics for people to appreciate the importance of that. So I think the establishment of community advisory boards, the number of trials we've had, we have a much more aware public. And I think things like social media, and while it's a bit of a challenge to tease out what's fake news and what's true and factual, I think it's incumbent upon us to spend the time explaining things to the public. And in South Africa, an example is, you know, very early into the epidemic, we had the Minister of Health, who's been really, really a strong leadership and consistent voice, even before the cases started, on preparedness. Took a scientist with him on national TV, and you know the stuff about next slide, and we ran through a whole lot of slides, and the amount of communication that started to take place amongst peers using WhatsApp, Facebook, etc., was just stunning, and it underscored again the importance of communication. But the second is that sometimes people come to Africa to try and do a study quickly and on the cheap. And one of the things we need to be mindful about is standards of care or standards of prevention. So certainly, you know, if you want to do a study about COVID-19 and spread it, you can't simply come in with vaccines without the protective gear for the study team or for the individuals, etc. So communication is important. The way we do these trials shouldn't be about quick and cheap, but we have to understand that and be respectful of the populations that we include in our trials. And I think when we all part of this partnership, the scientists, the collaborative relationship, the partnership, and that's what I think about in terms of partnerships, is where it's not you coming in and making me, an African scientist, a specimen collector or a data collector, but respecting my scientific contribution, and also respecting, as I respect the communities I work in. And I think when we can have those three aligned, then we really can move towards success in a very ethical way, in an effective way, and efficient way.
Indeed. I mean, designing, running a good clinical trial, it is very complex. And Tim, one question that keeps coming up is why will it take so long to develop a vaccine? Is there anything, looking at technology today, anything new that will give us a vaccine before we celebrate, let's say, the next new year, 2021, before December? Take us a little bit, you know, behind what is happening in the world of vaccinology.
Well, thank you, Otto. And Quarraisha, thank you. You really set the stage very well for a discussion about vaccine development and what it means for the continent of Africa. So let me start off by explaining a little bit about vaccine science and how much we've learned. I think it's fair to say that vaccines are one of the greatest health interventions we've ever had. And it's remarkable to think about how much disease and morbidity and mortality have been prevented by the very safe and effective vaccines we have in the world. So let's look at the coronavirus. Now, the goal of a vaccine they're designed to stimulate the human immune system in uninfected people so that it will provide protection when exposed to the virus. So first we'll look at how does coronavirus infect humans. So shown on the top is the virus. You've heard a lot probably about the spike protein. Now the spike protein is analogous to the key that the virus uses to unlock a lock to get into a human cell. And on the human cell, it's the ACE receptor that the spike protein connects to that allows the virus to enter a human cell. It's an RNA virus, the nucleic material. That nucleic material enters a vesicle, and then the virus does its business. It starts generating virus protein. It assembles new virus particles, and then those viruses are released, and that's the initiation of infection. Next slide, please. So the human immune response, when we get exposed to a virus, there's an immediate reaction to having viruses enter our body because they're recognized as not being part of the host. So there is something called these antigen presenting cells that will engulf, they sort of eat the viruses, and then they work with T helper cells. And the T helper cells stimulate two parts of our immune system. One are called B cells. Now, B cells produce antibodies. So that's an important first line. This can happen quite quickly, that B cells that the T helper cell has stimulated, they begin to produce these antibodies shown here as these little blue Y-shaped figures, and they actually attach themselves to the virus. Hopefully they neutralize the virus so it can't do its bad business, and you can stop an infection with enough neutralizing antibody. The other arm of the immune system is the T cells, cytotoxic T cells. Now they actually are designed so they can recognize a human cell that's infected with the coronavirus and they can destroy that. So the vaccine may stimulate B cells to produce protective antibodies or T cells which can destroy infected cells. And both of these cells are long living. Once the infection is controlled, for the vast majority of people, coronavirus infects them, the immune system kicks in, produces antibodies, produces T cells, it controls the infection, and over the course of perhaps 2 weeks, most people clear the infection and they move on. However, some people don't control it well, they get very sick, and we see a mortality rate of at least 1% with COVID disease. So what a vaccine is trying to do is stimulate that immune response. So you give it to a healthy person, you already have B cells and T cells that are aware of this virus, and then they're prepared once they're exposed. Next slide, please. So Otto, in answer to your question, the vaccine science has moved extraordinarily fast over the last many years. Now, as you know, we still do not yet have an effective HIV vaccine, and that's because of the special nature of the HIV virus that enters human cells and hides in our cells. But I'll go through this quickly. There's a number of different types of vaccines. The first column are DNA or RNA vaccines. Now, these are relatively new. They're easy to develop because once you have the genetic sequence of the virus, you can create DNA or RNA that mimics that virus. So the good news is they can move very quickly. However, they've never been done before. We do not have any vaccines on the market that are DNA or RNA vaccines. So it's exciting science. We hope that it's safe, but we're moving forward quickly with a number of these products that I'll talk about in my next slide. Another way to vaccinate is you actually attenuate a virus. So you have a live coronavirus and you inactivate it so it can't be fully active, and it stimulates the immune system, and that's another way for stimulating immunity. Otherwise, you can actually inactivate the virus so it's no longer live, but it stimulates the immune system. We think that's not quite as effective in terms of the immune response, but that's the way polio vaccines work, are to use an inactivated polio virus. And then there's subunit vaccines. The spike protein is a popular antigen that's used, and you take you synthetically make copies of the spike protein, You use them as part of the immunogen. This is the way we work with hepatitis B vaccines, pertussis, and HPV vaccines. You show the immune system a piece of the virus, and then you stimulate an immune response that can recognize that when you're exposed to the actual virus. Another popular method that's used is using a viral vector. You take something like adenovirus, which is a cause of the common cold, and you put part of the coronavirus in a relatively innocuous virus like adenovirus. And this is the way the Ebola virus vaccines have been created, and some others in veterinary medicine. And several of those are moving ahead quickly. Next slide. So this is another way of looking at the platform. The DNA vaccines, the Inovia vaccine, which is in human trials, is a DNA vaccine. The RNA vaccines are also moving forward. Moderna is one of the companies that's farthest down the track in the United States, and they anticipate starting a phase 3 trial this month. Imperial College also has an RNA. And if you were watching the news yesterday, Pfizer, the large pharmaceutical company, has a messenger RNA vaccine that showed promising results and is moving forward in human trials. Sinovac in China is making an inactivated one. Live viruses that are attenuated are shown here, the Serum Institute in India. Non-replicating vectors, these adenoviruses, Janssen, the Oxford product, which is done in conjunction with AstraZeneca. And then there's several protein subunits shown here, GSK, Sanofi, Novavax, etc. A measles vector vaccine, and then something called VSV, vesiculostomatitis virus. So several different approaches. As Quarraisha showed a slide that there are more than 120 products. So we're going to take a lot of shots on goal with this, and at least 10 products are in human trials that are moving forward. Next slide. So this is an unprecedented situation. We really have never had an epidemic like this that's swept around the world in 6 months. We have at least 10 million infections diagnosed, but we think there's far more than that because many go undiagnosed, and at least 500,000 deaths so far. So because of the nature of the coronavirus, which has shut down economies, and that's never happened in the history of humankind, where actually whole economies around the world have been shut down to stop the transmission that happens between people. So because of that, the vaccine development and evaluation process has gone into very fast moving forward. So we're in a phase now where there's the clinical development or the preclinical development in laboratories that's happening in hundreds of labs around the world. At least 10 products have moved into human trials. A key issue though for vaccines is safety. And we'll talk about that in an upcoming slide. It's imperative that the vaccines are very safe because first of all, we have to have vaccines that are safe as well as effective. And Quarraisha touched on the issue of the acceptability of vaccines. And if we lose confidence in the population that these vaccines are safe, it'll be very difficult to deploy a vaccine. So what's happening in the course of a pandemic outbreak is the science that normally would have taken in steps. You do preclinical, you do clinical, you do an efficacy trial, and then you begin to ramp up your production capacity. That's been moved forward. So now manufacturing development, scale up, and all the ability to mass produce vaccines, which is no easy thing to do. It's one thing to make enough vaccine for a trial. It's another thing to be able to make vaccines for hundreds of millions or billions of people. So there's already investments now in manufacturing and large scale capacity that's moving forward before the trials are even done. Next slide, please. So I have 2 slides just to talk about vaccine evaluation so we all have the same fundamental understanding. A great deal of work on preclinical laboratory work and then into animal studies. Often non-human primates like macaques or other monkeys are used to test vaccines to see if they can provoke an immune response. And then after there's really solid data on preliminary safety in animals and some efficacy, we move into human clinical trials. Phase 1 trials, the primary purpose of a phase 1 trial is to assess safety. We really need to make sure that the vaccines that we give people are safe, and they measure the immune response in a relatively small number of healthy people. Usually these do not include the elderly, they do not include children, so it's healthy, relatively young adults, and it may be 10 to 50 people in a phase 1 trial. If those go well and it appears safe and you get good immune response, you move into a phase 2 trial. Again, safety is always foremost in vaccine evaluation. You get further information on the immune response. You may determine what the dosage is. Often there's a vaccine schedule of more than one vaccine, and is it better to give them 1 month apart, 2 months apart? And the route of administration, are they intramuscular, subcutaneous? What's the best way to do it? And phase 2 trials might have 100 people, maybe 200 people. So if those go well, you have a safe product, it's generating an immune response, you think you know the right dosage and the right schedule, then you would embark upon a phase 3, which is known as an efficacy trial. And that's, again, you have to continue to assess safety, which always has to be foremost, but it's really the protective efficacy. And a phase 3 trial is in the thousands. The early phase 3 trials for coronavirus, for COVID-19, will enroll about 30,000 people at risk of infection. They're usually placebo-controlled trials, so some people get the active vaccine, some get the placebo, and you compare the infection rates in the two groups to see if the product, one, is safe and also can protect from infection. Now, the endpoints in the trial may vary. Ideally, you'd like something called sterilizing immunity. It means it totally prevents the infection from getting hold in people. Another endpoint might be a vaccine that doesn't totally prevent infection, but it dramatically decreases the severity of illness. So nobody gets very sick. You might get a mild infection, and so you get an immune response both to the vaccine and to the mild infection that you've had. So it's important to assess vaccines when you do efficacy trials, really at all ages. We know that the elderly people have an impaired response to vaccines. They often don't respond well to pneumococcal vaccines or influenza vaccines. And we also will need to assess vaccines in various populations and geographies. The coexistence of other comorbidities, whether that's HIV infection, tuberculosis, other conditions, underlying health conditions that might interfere with vaccine responsiveness, needs to be taken into account. And also how children are dealt with is a very special situation. Often children are not included in large trials. There's a demonstration of efficacy in adults, and then bridging studies are done to deal with children. So I have just one more slide. And again, this has to be foremost in the mind on vaccine development. Vaccine safety is really critically important. It would really be an unfortunate misstep if a vaccine was evaluated without adequate safety evaluations and then was deployed and was demonstrated to have serious problems. Now, in the past, the history of vaccines, some vaccines have caused something called antibody-dependent enhancement, so that the antibodies provoked by a vaccine could actually do harm and could make the disease work. We see this in dengue fever often, that often the second infection with dengue is more severe than the first because the pre-existing antibodies cause a more serious disease. And it's been an issue with dengue vaccine production of making sure that a dengue vaccine doesn't provoke this more serious disease. So for COVID-19, where we would be moving forward to immunize ideally billions of people, most of whom are quite healthy, you really want to avoid serious adverse events giving a vaccine in healthy people. So just think, for example, if you had a vaccine that only 1 in 1,000 people developed a serious adverse event. That may sound like a small risk, 1 in 1,000. However, if you immunized 100 million people, you would result in 100,000 serious adverse events. So these would be of great importance. Serious adverse events need to be avoided really at all costs, and early trials need to assess this. And it's some balancing acts we're going to have to take on, because the first trials will probably have data on about 30,000 people. So it may be hard to assess serious adverse events that occur at a smaller rate than this. So we're going to have to be prudent in how we move forward with trials, keeping safety at the front of our minds, and moving forward really in a deliberate, balancing the benefits of vaccines and the potential risks, both taking safety into mind, and as Quarraisha pointed out, a really strong communication message needs to go forward, and we really need to work collaboratively so that the populations we're working with have trust in the vaccine manufacturers and the public health officials deploying them. So Otto, I think that's my last slide, and happy to answer questions and have a discussion.
Thank you very much, Tim. And I think, you know, the word of caution and prudence is a good segue in the next question that I wanted to pose to both of you, in the sense that there are also a number of people who are looking around and say, is it really true that you're going to deliver on time? We've been after the HIV vaccine for 40 years, and we still don't have any vaccine right as yet. If you look at the seasonal influenza vaccine, it's partially effective. Every year we have to develop one. So what makes us so optimistic about it? So billions of dollars, a lot of head of states are behind this commitment to get to the vaccine. So there's also a little bit of politics that is involved. We know that this on the science side it is. So we need to be really clear with the populations. What is it to expect? Because the moment we start saying, okay, the clinical trials have started in the US, in Europe, in China, and in South Africa as well, the hopes goes high. So FDA has set a threshold of efficacy of 50% for approval of any candidate at this point in time. What does that translate to?
Yeah. Well, let me take a first crack at that, and then I'm sure Quarraisha will weigh in on this issue. You framed it very well. We're in an unprecedented pandemic with great anxiety, concern, economies have been shut down, and there really is a balancing act between a public health response, which limits disease transmission, and an economy that wants to get going, that requires people to be in factories and people going about their business, which puts them at risk of transmission. So everyone's really hopeful that a vaccine can allow us to get back to the good old days that you described at the beginning, Otto, 6 months ago, when we could have our normal lives. Ideally, a safe and effective vaccine would allow us to do that. However, the steps getting to deploying a safe and effective vaccines are quite considerable. So what's the likelihood of having success? I think there's a number of good signs. That the good news is, for the vast majority of people that become infected with the coronavirus, the SARS-CoV-2 virus, they mount an immune response, they clear the virus, and they don't get very sick. So that demonstrates that the immune system in many humans can quickly see the virus, handle the virus, and then clear it. How long that immunity lasts, we don't know yet. So that provides some optimism that a vaccine will be able to simulate that interaction with the vaccine product and the immune system to stimulate an effective vaccine. There's early animal models that show optimism. Early human studies with antibodies that are called neutralizing. They can inactivate the virus, but those don't translate into efficacy when in the real world. So I think we need to temper that sometimes exuberant optimism, often provoked by politicians, that we're going to be having one by the end of this year or early next year, with really a good, deliberate scientific process that keeps safety foremost in mind. We're moving very quickly. I have great confidence in the scientific process. The groups developing the vaccines are very good. We have great clinical science capacity to do large-scale trials. People know how to do these studies. There are large networks moving forward. As you mentioned, it was wonderful to see just in the month of June that the first human trial started in Johannesburg just a couple of weeks ago, with one of the products being developed in the UK, and really around the world, in Europe, North America, China, India developing products, and we very much want Africa to be fully engaged in the scientific process of moving forward. So the deliberate steps of phase 1, phase 2, phase 3 need to take place, and the safety and efficacy data will yield from those. We do need to temper sort of over exuberance in people that are saying, well, now we have a vaccine, we can get back to normal. Even if we had a safe and effective vaccine early in 2021, scaling up the number of doses required would take some time. That's why having multiple products would be very important. So it's going to be a balancing act, I think, between the optimism of a safe and effective vaccine and the realities of getting it out so it could really protect populations. Quarraisha, what do you think?
Yeah, I think, Tim, you've covered some of the critical issues. Just to add perhaps a few more points is we have a lot of candidate vaccines, and we have a few that are entering clinical testing. But in our experience with other epidemics and other infections, and doing clinical trials have introduced a certain amount of realism into this. So firstly, when we're doing a trial, we really don't know whether something is going to work or not. Mm-hmm. And these early phase studies are really about safety. It's really when you get to the bigger studies that you're getting to, does this work? Because you need both safety and you want something that works. And a 50% efficacious vaccine is not a shabby target, because if you get that, and with coverage and large coverage, like when I spoke about the non-pharmaceutical interventions we have, it's the coverage that really gives you the protective benefit. But my concern is, and not a concern, but more like the reality check, is that as clinical trialists, we often have the label of being experts at failing, because when we do these studies, 9 times out of 10, if not more, we don't get the result we want. Although we expect and we gave it the best shot, these trials are done because we don't know, and it's always unpredictable what that outcome can be. And we've certainly learned that extensively from HIV, from TB, from malaria. The things that we think are going to work don't, and that's why we need the trials to do that. Secondly, I think when we think about COVID-19 and we're seeing the rate at which people are being infected, there's a lot of immunity that's being established in countries and settings where the magnitude and the spread has been rapid. But we're also learning that that natural immunity is not sufficient. Within that, we know some populations are more vulnerable, and we think, for example, about our first responders, particularly healthcare workers, but even people involved in other production sectors are critically important, and you want to make sure that they're not under any threat. But the other groups are and we've seen age is really important, is older populations. And most countries, even if they have a stay-at-home policy, the message that's going out is that protect the elderly in our communities. And so you've got this balancing act where when you have a vaccine and say you want to protect the elderly, they're also in a very contained environment. So to know something works also requires exposure to the virus post-immunization. And when you exposed to wild-type virus, will be able to answer the question, will it work or not? So 3 things, a virus that comes and spread within days. So timing is really important for vaccine trials, because you blink your eye and it's gone and through. And there are few countries, and Africa being one, where you're still at an early stage and becomes more and more important for vaccine trials. So speed of the disease. 2 is who are we going to prioritize for vaccine trials? And there's vulnerability in terms of age, but there's also, I think, while we have a young population in Africa that also is a plus for vaccine trials, that we do have a lot of comorbidities in Africa, and we're seeing increasingly things like obesity and diabetes and hypertension are featuring quite strongly in terms of disease severity and deaths. And so it's this, I think Tim said it nicely, it's a very fine balancing act. It's almost like a sort of art and science in terms of how we select candidates, the timing of these trials, who we evaluate it in, and how we evaluate it is going to be, at the end of the day, that mix is what's going to determine over and above everything, the best chance of success. But even with all of that, science, and that's the beauty of it, it's like this elusive thing. Every time you think you have it nailed, you have one more challenge coming in. While it's fun being a scientist, but trying to do this in a pandemic where, you know, it's a state of emergency we're in, it's how do we juggle all of these different factors? You know, we've seen, for example, we have lots of experience with vector-based vaccines, but not too much with, we don't have a single RNA vaccine. So while it sounds exciting and promising, it's a precedent again. But my optimism comes that we have set amazing precedents, unparalleled in our history of being human beings, that we will find something, and we'll find something fast. And so while there's a lot of lessons to be learned from the past and what we've learned to date, that's my source of optimism, Otto.
Oh, thank you so much, both. I mean, with every exciting and good conversation, you know, time does not cooperate with us. So we're quickly running out of time, but I still have just one question before we close this good conversation, great conversation. If you can, in the 30 seconds or 1 minute, tell me, because this is not going to be the last pandemic we see, and it's going to hit the global south again, just, you know, COVID-19 has. We had HIV, we had Ebola yesterday, and we're learning as we go. We're becoming those experts in failing, right? How much have we built, and how can we take advantage of this investment in COVID-19 to build in preparation for the next pandemic? 30 seconds, 1 minute.
I'll just jump in. Oh, Quarraisha, go ahead.
Go. I was just going to say in 10 seconds, Otto, absolute right question. And Africa's resilience has come from the investments, the public health investments in responding to HIV, TB, malaria, Ebola. And I think that's the difference and the contrast that we're seeing with COVID-19. But we still need to build much more capacity than we already have. So while we have that advantage, it's not enough. We need to invest more to be even more resilient. And they say this coming century is going to be splattered all the time with different epidemic challenges. So this is a good time for us to start to make those investments. Sorry, it took more than 10 seconds.
Tim?
Yeah, no, just following that, I think, you know, we talked about, you know, for my whole global health career, we talked about the big one, you know, would it ever come? And we failed to prepare for the big one because we're living the big one right now. So I think this is the global wake-up call. We need to make investments in our global public health system, the ability to respond to these diagnostics. Hopefully the vaccine science will move us forward. We'll learn from these trials. So hopefully we'll be able to put COVID-19 somewhat under control in the not too distant future. But as you appropriately point out, Otto, it's not going to be the last one. We need to learn more about emerging pathogens, be prepared for them. But I'm most encouraged by the young scientists that we see around the world that are enthusiastic. They will come up with new ideas that'll move us forward. So we need to seize this moment, continue developing a global workforce, make the investments, and be smart working together for the next epidemics. Thank you.
This has been one of the most enlightening conversations I've had in a long while. Thank you both for sharing generously your thoughts and experience. I remind everybody that this was a conversation with Quarraisha Abdool Karim, Associate Scientific Director of Quarraisha, and Dr. Tim Mastro, Chief Science Officer at FHI 360. Thanks for joining me today. I wish you both good tidings in these extraordinary times. Thank you so much. Until we meet again. Bye-bye.
Thanks, Otto.
Thank you.
Bye.
Thank you, Ada. Take good care, Quarraisha. Good to work with you today.
Thanks. Bye.